How's
There you
that?
go. Yep.
Okay. When I tested it before, it worked fine, but that's part of this process. So thank you, Phil, and thank you, Cori, for having us here. And Vic actually invited me to give the Virology 101 piece of this because I do teach the 400 level virology class in the microbiology and molecular genetics department, and I've been doing that for 3 years now. So our talk today will begin with a primer on virus biology. And then I'll talk a bit about coronavirus biology, and then wrapping up with a discussion, a brief discussion of some of the drugs that are in clinical trials right now to fight the COVID-19 disease. So after that, Vic will pick things up talking about the fundamental principles of vaccination and the different types of vaccines. So when we think about viruses, most of us think about the viruses that are listed here. We've all suffered from viral infections. Some are short-lived and acute, and others are more serious, cause more serious diseases and can even be life-threatening. We are protected against some of these viruses by vaccines. I did want to take a minute, though, to talk about how viruses do not just infect humans. In fact, viruses will infect any type of organism or any type of cell. If there is a cell type out there, there are viruses that can infect it. And what we're looking at here are some examples of viruses that have a significant impact on agriculture. The foot and mouth disease virus causes lesions in the hooves and mouths of cattle and livestock and cause them not to be able to eat or even be able to stand sometimes. You see a picture here of fowl pox, which is the chicken version of chicken pox. The deformed wing virus can cause devastating impacts on beehives. So we can see this is just a sampling of the types of viruses that impact animals besides humans. Viruses also infect plants. The variation in color that you see on these tulips is the result of tulip breaking virus. The plant is infected with tulip breaking virus. These cucumbers are infected by cucumber mosaic virus. And then here in the lower panel, you see the impact of tobacco mosaic virus on orchid leaves. Tobacco mosaic virus is a plant virus that infects a variety of plant types. And actually, Roger Beachy, who is a member of the Dean's Board of Advisors for the College of Natural Science and who obtained his Ph.D. at MSU, is an expert in plant virology and has used TMV as a model for many of his studies. In addition to plants and animals, viruses also infect single-celled organisms. So viruses that infect bacteria are called bacteriophage, and we can see an example of those here. And then also viruses can infect things like amoeba. This virus shown here is called Mimi virus. It's a huge virus that has, we have a lot to learn about it, and it is made by the single-celled eukaryotic organisms that are amoeba. So those are the types of places that we find viruses and the types of things they infect. And so now I wanted to take some time to really define what is a virus. How do we go about defining a virus? And so the first thing we want to consider is that these viruses are small, infectious, obligate parasites. And what that means is they need to be inside a cell in order to replicate. The next thing we want to think about is the genome. The genome can be made of either RNA or DNA. Viruses hijack the cellular machinery to make new virus parts. They cannot, on their own, make those virus parts. They do contain the information necessary to direct the cell to do so. And then eventually you'll get new virus particles that are formed from these newly synthesized components. That new virus particle, or virion, is the infectious agent that is used to transmit the virus to the next cell or to a new organism where this cycle begins again. So if we think about that definition, we want to create a model of a virus. And the first thing in that model is the genome. And that genome, like as I said in the previous slide, can be either RNA or DNA. This is different than our genomes, which are DNA. You can't have, humans do not have RNA genomes. Then the second piece of the virus is a protein coat called a capsid. And the protein coat will surround the genome. And this, in many cases, is all you need for a mature virus particle. Some viruses will add to that a lipid envelope, and this is basically a lipid bilayer that surrounds the capsid, which is enclosing the genome. Depending on the type of virus, there will be other viral proteins included in the virion particle, but some of them, it's just as simple as one copy of a genome and then several copies of one or more capsid proteins, and that's all you need. So the simplicity of these entities are both beautiful and nefarious, in my opinion. They have genomes as small as 8,000 nucleotides or less, compared to the 3 billion nucleotide long human genome. And yet they can come in and completely hijack a human cell and cause significant morbidity and mortality. The other thing that's remarkable, given this level of simplicity, is the diversity. We already saw an example of that diversity in the different types of organisms that viruses can infect. So given that diversity, it's important to have ways to classify viruses. So I'm just going to take a couple moments to talk about how viruses are classified. We can look at the different properties. First of all, you can look at the morphology, the size, the shape, the symmetry of the capsid. We also talked about whether or not a virus might have that lipid bilayer, that envelope on the outside of the virus. That is an important characteristic. Then we think about the specific proteins. What are the function of the structural proteins, those proteins that make up the virus particle? What are the function of the non-structural proteins? Those are important for viral replication. And we can look at the amino acid sequences of those viruses, of those proteins. All of those things in turn will determine the biology of the virus. What's the host range? What types of organisms does it infect, and what types of cells within those organisms? How is the virus transmitted? What is the pathogenicity? But the overarching characteristic is the genome. So if we know the genome sequence, we can start to predict all of these other characteristics. We can figure out what the proteins will be, and therefore how the biology of the virus is determined. We can think about the structure of those proteins confers the shape of the virus particle. So those are the types of things we use to classify viruses. One of the most useful parts of that classification is the genome. And the Baltimore Classification guides researchers in thinking about how viruses replicate. The Baltimore Classification is dependent on what is the type of genome in terms of the type of nucleic acid, and whether or not it is double-stranded, single-stranded, and the polarity of that genome. So I'm just listing some examples of viruses that fall into under these categories. We know a lot about how a virus replicates just by knowing what the Baltimore Classification is. So DNA viruses can be double-stranded or single-stranded. Examples include human papillomavirus or the B19 virus. The viruses that we most likely or most often hear about are RNA viruses, like the coronaviruses. Those can be double-stranded RNA with 2 strands of RNA, or we can have single-stranded versions. Rotavirus is a well-known virus. Rhinoviruses cause cold viruses, and we've all heard of influenza virus before. The third group in the Baltimore Classification are reverse transcribing viruses. And these are viruses that contain the reverse transcriptase enzyme, which can synthesize RNA from, or DNA from an RNA template. And so HIV is the most well-known retrovirus that falls into this group, and also hepatitis B has this kind of intermediate. So how do coronaviruses fit into this classification? Well, thinking about those characteristics in the Baltimore Classification, we know that SARS CoV-2 is an enveloped plus-strand RNA virus. And what you're looking at here is an electron micrograph of virions isolated from a patient in the United States. And then we have a cartoon version of what you're actually looking at in that electron micrograph. We have a single copy of the plus strand RNA genome that's surrounded by several copies of the nucleocapsid protein. Surrounding that then is the lipid bilayer, which is that envelope that I talked about. Embedded in the lipid bilayer are these structural proteins, one of which you may have heard about in the news called the spike protein. There are several copies of this spike protein that are found on the surface of the virus particle, and it plays a very important role in replication. So that's the general classification based on the last couple of slides we talked about. We also have an organization called the International Committee on Taxonomy of Viruses that is responsible for putting viruses into particular groups and for naming viruses. And so I thought it was worthwhile to go over how this virus is named. So there's two different terms that we're hearing about, and one relates to the disease. So the disease is called coronavirus disease 2019 or COVID-19. So that's what the WHO named in terms of the World Health Organization in terms of the disease caused by the virus. There have not been 18 other diseases like this caused by coronavirus. The 19 is indicative of 2019, the year in which the virus was identified. The virus itself is called SARS CoV-2 or SARS coronavirus 2. It is very highly related to the SARS coronavirus that led to a pandemic in 2003. So that's how the virus has been named. And then we can think about the hierarchical classification of the virus as well. There's an order and a family, Coronaviridae, and then within that family, we have a couple subfamilies and then different genera. The alpha virus, or the alpha coronavirus genus does have viruses, human coronaviruses that do infect humans, but they cause diseases that are more like the common cold. The beta coronaviruses include the two SARS coronaviruses, as well as the Middle Eastern Respiratory Syndrome virus that you may have heard about, a virus that causes really significant morbidity and mortality. So that's a basic overview of how it's named and how it fits into some of these classifications. So now I wanted to spend a little time on how viruses replicate. The important thing for you to think about here goes back to your BioSci 111 or your BioSci 161 course if you're a more recent graduate, thinking back to how cells are structured and how they function. So we want to remember that a virus has to be inside a cell and it uses that cell machinery to make new parts. The other piece, going back to your old biosci courses and maybe biochemistry courses, is the flow of information. We want to remember that we have DNA that gets transcribed into RNA, and then that RNA is translated by ribosomes to make protein. In the case of coronaviruses, we're going to skip the DNA part because the genome is not made of DNA. It is in fact made of RNA. So what do those general phases look like? Well, first of all, the virus has to attach and enter a cell. You can't get a replication cycle until the virus attaches to a permissible cell and gets across that cell membrane. It has to cross that lipid bilayer. That doesn't happen easily. It doesn't just diffuse through. There has to be a mechanism for that to happen. Then once inside the cell, the genome has to be released. The genome is then going to direct the synthesis of new parts. One of the things that has to happen is that you make proteins. Some of those viral proteins will be responsible for making new viral genomes. Some of those viral proteins will be structural proteins that will end up in the new virus particle. Once you have enough new copies of genomes and new structural proteins, you will assemble a virus, and then eventually you have to release those virions from the cell. So that's the general overview of virus replication cycle. Now I'm just going to highlight a few of the specifics for coronaviruses. We could spend much more time on the details of this, and we did do that as we taught coronaviruses in the midst of the pandemic and MMG, the MMG virology course this past semester, but I'll just highlight some of the differences or important characteristics. So one of the interesting things goes to this attachment and entry process for coronaviruses. The SARS viruses actually have two pathways for attachment. Both of them involve first having that spike protein that I mentioned earlier bind to a receptor. The receptor is called ACE2. It's an angiotensin converting enzyme 2. That is a protein found on the surface of the cell. In one pathway, following this attachment, we have a process called receptor-mediated endocytosis, in which the virus is engulfed into this endosome structure. Within that endosome, thinking back to your chemistry courses, we have a pH change. So the pH change is going to cause some changes in the shapes of the proteins, and ultimately we will release the viral genome. And the other pathway, what happens is we're going to have fusion directly with the cell membrane. So this requires that there not only be the ACE2 receptor, but also a protein called a protease at the surface of the cell. The ACE2 is going to help bind the virus particle in the first place, and then this protease is going to cleave some amino acids in the spike protein, and that allows that fusion. So the envelope on the virus particle basically melts into the cell membrane because they're both lipid bilayers. And the ultimate goal here is the same as the other pathway. You release the genome into the cytoplasm. Once that genome is released, it looks like a messenger RNA, so it can be immediately translated by cellular ribosomes. So then we start to see viral proteins being synthesized. The first product is a long polyprotein, and that gets cleaved up into a bunch of smaller individual proteins. These form replicase transcriptase complexes that are responsible for making more genomes. We have to make a lot more genomes. Some of the RNAs are actually going to code for these structural proteins, like the spike protein and the envelope protein and the matrix protein. So we'll get a bunch of those made, we get a bunch of copies of our genome, and then eventually we'll assemble that into a mature virus particle. A virus particle will make its way via another vesicle to the surface of the cell, and then it will be released in a process called exocytosis, which is basically the opposite of that endocytosis we saw during the first type of entry. So this is just, as I said, a real quick overview. There's lots of details that go into this, but the key here, and the reason why I went over this, is that a drug or any other compound that interferes with any of these steps has the potential to serve as a therapeutic, as an antiviral. So thinking about that viral life cycle, we can look at some of the drugs in current clinical trials. You may have heard a lot about monoclonal antibodies or convalescent serum that has antibodies in it. These antibodies typically will bind to that spike protein on the surface of the virus particle. If the antibodies are binding to that spike protein, the spike protein cannot interact with the receptor on the cell surface, so the virus never even binds, and you don't even start the virus replication cycle. And second example is a drug called camostat mesylate. And basically what that does is it interferes with the second pathway where we had fusion right at the membrane. It inhibits that protease I mentioned. You have to have the protease activity, which is going to cleave the spike protein and allow fusion to occur. If you inhibit that protease, then the virus cannot release the genome into the cytoplasm, so you don't initiate a replication cycle. The third example that I have for you is chloroquine or hydroxychloroquine that you've heard about in the news. And the understanding that most researchers believe is impacted by this drug is this endosomal pathway. It interferes with the endosomal pH. And so if you don't have the right pH in there, you don't end up releasing the genome into the cytoplasm and a life cycle cannot ensue. And finally, the fourth example I have is of remdesivir, which is the most promising of these drugs. There are some others that have been shown not to be effective, but this is a drug that inhibits the RNA polymerase. So this is a really good candidate for a drug because it inhibits the RNA-dependent RNA polymerase in the virus. We, ourselves, do not have RNA-dependent RNA polymerases. We have no role in our cell for using RNA as a template to make more RNA. That is a virus-specific function. So drugs that inhibit these virus-specific functions and don't have an impact on cellular function are the most promising candidates for antivirals, not just for coronaviruses, but across the board. So that's a real quick version of Virology 101, and I'm going to stop sharing my screen, and Vic is going to pick up to talk about vaccines, as long as I can stop sharing my screen here.
Well, thank you, Cori. That was terrific, and I appreciate the introduction to viruses, because we're going to use a lot of that knowledge and apply it to thinking about vaccine production. I also want to thank Corey Palmer and Sarah Ford for helping set this up, and Phil for a great introduction. And also my friend Mark Ehlert, who I think may be here today, but he helped us really think about how to target this presentation today. And it was very useful for us to get Mark's input on that. So thank you, Mark. All right, I want to talk about vaccines from a lot of different contexts. We're going to think about how they're made, how they work, and sometimes why they don't work. And then we'll look a little bit about what we're doing in terms of COVID for vaccines. So the three things I want to get across right away is the question of how vaccines work very broadly. The whole idea of a vaccine is to stimulate the immune system to recognize a pathogen. That's one of the things that's the ultimate goal of a good vaccine, is to stimulate for long-lived immunity. That's not always what happens with vaccines, and we'll talk about some examples of that. And finally, the idea is that once we vaccinate somebody, their immune system has a surveillance mechanism to deal with natural encounter with a pathogen, which may occur 2 weeks later, may occur 2 months later, may occur 20 years later. And so how long-lived immunity lasts after vaccination is something that is an important point. Sometimes that changes not because of the nature of the immunity, but because of the pathogen itself, and we'll talk about some examples of that. I want to first, before we get into vaccinology per se, discuss some concepts and things you've been hearing a lot about, and they matter in terms of thinking about vaccinology for this pathogen. One of those is the concept of herd immunity. We've all heard about this quite a bit. Herd immunity is the idea that if enough individuals within a population or within a herd are protected from infection, the likelihood of transmission to a new person from an infected person is much lower. So let's look at two populations. In this scheme, we have two populations, this one and this one. And the map is that infected but not vaccinated people are in red. Healthy people who are not vaccinated, in other words, they are the susceptibles in the population, are in clear or white circles. And then the healthy but vaccinated people are in blue. So if you have a population such as this one on the left with a lot of not vaccinated healthy people who are not infected either, so the white circles, and you do not have many people who have been vaccinated, the blue circles, the likelihood for an infected person to encounter an uninfected person is much higher than it is in a population like this, where we have a lot of healthy vaccinated people circulating in the population. And so the likelihood for a virus from one of the red infected, not vaccinated individuals to find a healthy, not vaccinated person who's not infected is very low. So the more vaccinated or immunized people that we have in a population, the less likely it is for a virus to go from an infected person to a susceptible person. And so that's the concept of herd immunity, and that can be generated in two ways. It can be generated by vaccination programs where we actively infect or inject people with vaccines, or it can be achieved through natural infection, and that usually takes a lot longer. And we've heard estimates of how many people need to be infected, up to 60 or 70% of the population in order to get and convalesced in order to get good herd immunity. So this is a feature that occurs on the basis of the transmission of the virus, the likelihood of identifying uninfected people, and the quality of the immunization of the individuals who are protected. All right, another concept that we need to think about is the terms antigens and antibodies. All right, so this is very much immunology 101. An antigen is a particular substance that stimulates an immune response in a person. So this is really what the vaccine that we would give somebody is. It's an antigen. It's a part of a pathogen, or it could be the actual pathogen in a natural infection. And the antigen stimulates antibody production by the immune system, and those antibodies are typically directed very effectively against that specific antigen. So in a bloodstream that has antigens with antibodies also in the bloodstream, there will be antigen-antibody complexes that form because the antibodies have high affinity for binding the antigen, and those can be cleared in the liver typically. And once those are cleared, then the antigen has left the body by being taken out by antibodies. And so that's the goal of vaccination, to develop a high level of antibodies that recognize a specific antigen. All right, so let's look at what happens when we get an immune response to an antigen. This is a graph that shows on the y-axis the magnitude of an antibody response, just in a log scale, and on the x-axis, the number of days after exposure of the person to the antigen. And the antigen that we're looking at is in this blue graph, antigen A. So on day one, we expose a person to antigen A, and what happens is there's a noticeable, detectable antibody response that is not very long-lived. Within a few days, it reaches its peak, and it's not a very high peak, and then it wanes over several days to the point where it is no longer a particularly aggressive antibody response. However, the immune system in this process, and we'll look at that next, has been trained so that the next time this antigen A comes into the system, the response is a lot more robust, a lot more aggressive, high levels of antibodies are produced, and in fact, the quality of those antibodies is better, more able to bind the antigen, better affinity than the original antibodies. So the secondary immune response is very aggressive, and also, as this graph shows, very specific. So if we introduce a second antigen at this point, that antigen has to be seen by the immune system for the first time exactly the same way as antigen A did the first time it was seen by the immune system. So what we're trying to do with vaccination essentially is to get this established so that when the pathogen comes in as the second exposure, we get this aggressive, long-lived, productive response. So the process of directing immunity against the pathogen then is what we call immunization. I want to look a little bit about what's happening in the immune system when this occurs, because that'll help us think about some of the vaccines that we're seeing developed against COVID. So we'll look a little bit more at the cellular level. Essentially, so on this side of the figure, we're looking at the primary response, and on this side, we're looking at the secondary response. And this is just a graphic to show us roughly what's happening in the immune system, and in particular with two immune cell types that are important in vaccine development or in immunology in general, and that is B cells, which are produced in the bone marrow, and these are shown in purple, and T cells, which are produced in the thymus, and those are shown in green. B cells are essentially the antibody producers. They are the cells that produce antibodies and do so very effectively after they've been stimulated by an antigen. T cells have really two functions in the immune system. One function or set of functions for T cells is that they are regulators. So they control a lot of the outcomes in the immune system. They help other cells do their role very well, and they also produce regulatory molecules that control some of the cell types that are brought into the immune response. They also can be killers. So T cells can be activated to kill infected cells in our body before those infected cells can release virus and let other cells become infected. They can also be trained T cells to kill cancer cells, and T cell cancer therapy is a very active form of research and development for cancer immunotherapeutics. So what happens in the primary immune response? We think of the antigen in this case as bits of a virus or a bacterium. We haven't talked anything about bacteria, but of course there are a lot of pathogens that are not viruses but are bacteria, a lot more complex cells. Let's focus on viruses. So bits of viruses are recognized in the primary immune response, and these could be from the natural infection or they could be from a vaccine. The naive B and T cells recognize those bits of the virus and are stimulated to go down the developmental pathway or mature, and they mature into what we call memory cells, and the whole idea is to establish immune memory so that in the secondary immune response, the immune system remembers, if you want to put anthropological terms on it or anthropomorphic terms on it, the immune response remembers that it has seen this antigen before, and now in the secondary immune response, these memory B cells and memory T cells expand to very high levels. So now we have memory B cells that are expanding, so we have many more of them, and they are producing highly specific, high affinity antibodies against the original virus the next time it sees that, the next time it's exposed to that virus. So the same thing happens with T cells. They expand. Memory T cells that expand can become killer cells to kill infected cells. They can also regulate different aspects of the immune response. So what we go from is a naive immune system with the first exposure to a very highly trained, highly aggressive, highly specific immune response with the second exposure. So in vaccinology, we're thinking about getting this going with the vaccine and then having this happen when the exposure happens down the road when you actually see the pathogen. Okay, we actually had a question about what happens when something mutates, what actually mutates, and how does that lead to changes and differences and concerns for vaccine. This is something we deal with and you've dealt with every year. If you get a vaccine for flu every year, which I hope you all do, and it really, honestly, I expect that you all do. Why do we need a flu vaccine every year if what I've been saying is we get this nice long-lived aggressive immunity? Well, there are a lot of reasons for that, a lot of reasons why immunity might not be so great for a long period of time. But in this case of flu in particular, the reason for that has nothing to do with the quality of the immune response. It has to do with the virus. And what happens with flu and other pathogens as well, but we'll focus on flu, is that over time, the circulating virus that's in our population acquires mutations. And if the original vaccine is directed against, in this cartoon, virus A, and the antibodies that the original vaccine led to in the vaccination targeted the triangle protein and the circle protein on the surface of this virus, could be a spike protein and a neuraminidase or something, it doesn't matter. But in this cartoon, our effective antibodies with vaccine in 2020 direct against a triangle and a circle. And in 2021, we get a new virus circulating in the population where mutations have led to the triangle going away. And now we have a rectangle on the surface of this cell. It might have altered a protein in a subtle way that is still functional for the virus, but is no longer a good antigen for that vaccine that was developed in 2020 vaccination. So we need a new vaccine in 2021 because the virus circulating in the population is encountering a population that has antibodies against this protein, but not against this protein. And those immune responses, those vaccine responses, aren't going to be as good against this virus as they are against this. There are a lot of pathogens that make a living out of changing their surface structure so that the immune system cannot recognize them continually. A classic example for bacteria is the sexually transmitted disease gonococcus, the gonorrhea or produced by the bacterium gonococcus, and that bug changes its surface continually, and so we have yet to develop a good vaccine against that disease. But that's what happens with flu vaccine every year. We have to change the formulation to keep up with what the virus is doing through mutation. All right, now there's different types of immunity. I've been talking about active immunity, where we inject somebody with an antigen and we develop antibodies, or we get a natural infection that leads to protection down the road. That's active immunity, where the patient or the individual is developing her or his own immune response that will recognize the pathogen when it needs to be recognized to clear for the person to survive. There's also passive immunity, and Cori touched on that a little bit. We can take antibodies from people who have become infected and give those antibodies after we purify them, or we can construct antibodies in the laboratory, a specific type of antibodies called monoclonal antibodies that will target the pathogen if we give them to people. A classic natural example of passive immunity is breastfeeding, because mothers who breastfeed their children are passing nutrients through the breast milk, but are also passing antibodies that will protect that child from a lot of infections until their own immune system is developed enough to start making antibodies on their own. So passive protection by maternal antibodies is a classic form of natural passive immunity. And then artificial passive immunity relates to using convalescent sera from people who have been infected. Now at Michigan State, we have some high credit, I think, to take nationally on the COVID convalescent plasma project. This is the leadership of that project, which is a national group of individuals who are looking to identify convalescent sera as a potential therapeutic. It's being used in people. Our Nigel Paneth, who's in the Department of Epidemiology and Biostatistics here in the College of Human Medicine, is one of the leaders of this national organization to identify donors for convalescent plasma that will be used and is being tested for use in acute situations such as healthcare workers or people who are infected and are rapidly losing health. And so people on ventilators have been given these convalescent plasma. So this is an ongoing project. It's exciting, and it really is has Michigan State leadership all over it. So that's wonderful for all of us, I think. All right, what are the different types of vaccines that we think about and that we use? There are really several forms, and we've all been infected or we've all been inoculated with multiple ones of these, because we've gotten so many vaccines over our lifespan. Live attenuated viruses in particular are often used. So these are pathogens that have been disarmed in some way, so they can no longer cause disease, but they are still able to replicate in the body and induce an immune response. And classic examples of these are measles, mumps, rubella. These are all childhood vaccines that are given to kids. These are live viruses, but they're avirulent. They don't cause disease. Same thing can be done with live viruses for vaccines, but they can be killed first so that they still maintain all of their antigens intact, and they can be given as an injection or as an oral preparation in some cases for killed vaccines. So these are the whole pathogen, but they're unlike the live ones. They're dead, but they still induce immunity. Toxoids, everybody who's listening to this talk has been vaccinated with a toxoid against diphtheria toxin. This is a bacterial toxin that causes a lot of damage. The bacterium itself is really not that bad. It's less of the concern than the toxin that it makes, which causes the disease. So we've got vaccines that directly target the toxin and are made of inactivated forms of the toxin called toxoids. And then subunit conjugates, the hepatitis B vaccine, one gene product, one protein of the virus is used as a subunit to inject, usually as part of a complex with some of these other vaccines, to stimulate immunity against that feature of the hepatitis B. Just a purified protein, or in some cases, we're starting to see the purified genetic material that's being used as a vaccine to deliver the genetic material so that the protein will be made inside of us, and then it will start inducing an immune response. So in fact, I want to just finish up with some of the coronavirus vaccine candidates that we're seeing. The one that's probably getting the most press right now, this is what Anthony Fauci talked about the other day in his testimony in Congress, is the Moderna vaccine, which is a nucleic acid vaccine. Cori gave us a great intro to Coronaviruses, these are RNA viruses. They're so-called positive strand RNA viruses. So the messenger RNA is the genome, and the Moderna vaccine that Fauci's talked about is a messenger RNA that encodes the spike protein, and that's just encoded by a lipid, and that's used to as an injection to give to people. That gene, essentially, that messenger RNA gets into our cells. It will begin to express the protein. It's recognized by our immune system, by antigen presenting cells, as a foreign antigen. It stimulates a T cell response that leads to all of the downstream effects that we get with T cell responses, including antibody production, T cell help, T cell killing. So it's a very effective vaccine or approach to stimulate broad immunity in our cells. There are a number of other, I think there's 161 vaccines that are being tested now in various forms. They include all of these different versions, protein-based purified proteins that are part of the capsid structure of the virus, attenuated benign viruses like adenovirus, into which we can introduce the genome or part of the genome of the coronavirus, so that that adenovirus will express an antigen that can then be delivered inside of the cell. So there are a number of different versions. Why does it take so long to produce a vaccine? And I'll just finish up with this. We have to remember that of all the therapies and all the things we give to people to protect them from illness, vaccines are practically, in terms of real medicines, I'm talking about medicine versus something like vitamin doses or things that we might take as nutraceuticals. But in terms of real medicines, the vaccines are the only ones we give to otherwise healthy people. So the process for producing safe vaccines is long and laborious and very precise. Research takes a long time. The preclinical preparation to make a batch that we can test in people, and then finding the right populations to test and deconvolute all the data that we get. These are blind tests of thousands of people that we're trying to determine whether the preparation is causing any problems in them, any health issues, safety and efficacy. Do we get an immune response? Is the immune response protective? So these things all take time. For COVID, we've thrown out the playbook, and these things are essentially, for many of the vaccines, certainly the Moderna vaccines, several of these processes are being done all at once. So we're already going into manufacture as if these other steps have occurred and they've been successful. So we're getting to step 5 essentially at the same time as we're doing step 2. This is highly costly, of course, because if it turns out that the clinical trials aren't working and millions of doses have already been made that aren't going to be useful, that's costly. So this is why it takes so long to get a vaccine, but we're accelerating this tremendously quickly now. So as Fauci said the other day, he expects people to have vaccines in them within the next 8 to 10 months, I believe is what he said. But again, the whole purpose of it is to stimulate immunity. And remember, always, these are going into healthy people. So we have to make sure that there are no issues with the vaccines when they go into those healthy people, other than that they stimulate immunity. So I'll stop there, and I think we're able to take some questions. I know there were many, and we can direct them to me or to Cori. I'd like to thank you all for your time and attention and for your support of the college, and thanks to Cori, and both Coris and Phil, etc. So for all of your assistance on putting this together. So I'll stop, and we can take any questions. Cori, I think you're ready to take questions as well. Am I right?
Yes.
Good. I think Cori Longley was going to run our show. Cori Palmer run the vaccine.
Well, one of the questions that we had, Vic, from one of our favorite alums, Ethel Cormier, said,
Ethel!
Yes.
How are you?
So are the rare second infections we are seeing reported based on a secondary response or mutated COVID-19 virus?
So I assume you mean, Ethel, the rare second infections with coronavirus. I think it's probably what you mean. So it's a great question. I don't, the early data or the early literature I saw on that, Ethel, was that there were some in South Korea, there were reinfections. I think that has been reinterpreted now as probably viral testing that was detecting fragments of the virus that were still present in the person, but were not a second infection. I think my sense has been that we still don't have any the long-term data on reinfection, but that it looks pretty good that people don't get infected a second time. Now, we're only 8 months into or less than 8 months. We're 4 or 5 months into this outbreak, so we still have a lot to learn. But I think the early data suggests that people are not getting reinfected. Cori, you probably have more knowledge about that than I do. I don't know if you've heard anything different.
I wouldn't have anything else to add to that. And I think a lot of the questions just require more data, and we won't know the answers to them for years to come in some cases. I've taught last year when I taught MMG 413 virology, I taught a unit on SARS when the topic was the 2003 pandemic. And we looked at a lot of the post-mortem studies, but it took years to really understand the spread and the transmission of the virus. We're collecting a lot of data, but I think it'll be a long time before we fully understand those kinds of issues.
Mm-hmm. Yeah, which is not to say, by the way, that, I mean, people might not get reinfected this year, but whether there's, like we say with flu, changes in the virus so that next year we have reinfection, that remains to be seen for sure. But at least right now, it looks like we're not getting acute reinfection.
You're still muted, Cori.
Sorry, I had a baby crying in the background. So another question related to vaccine is why is it that certain vaccines offer only several years of protection and require a booster, and others offer lifetime protection?
Right, that's a terrific question, and it's one that's an active area of research. And I hate to sound like, we should know that, but it is true what Cori says. There's still so much we don't know, even about vaccinology. That it is absolutely the case that when I was a kid, everybody got infected with measles. In fact, if you got measles and you had siblings, your mom threw the siblings in with you so that they would get infected, because a measles infection essentially provides lifelong protection against measles. So that immune response is obviously long-lived, very robust, very effective. That's not true for, we have vaccines against cholera, for example, which aren't particularly good vaccines, but the immunity that generates against them is also not particularly good. Now, why that is relates to the quality of the initial immune response, the cell types that are involved. In some cases, we didn't talk about anything related to something called adjuvants. We use adjuvants as essentially cofactors, for lack of a better term, that help stimulate the immunity to the antigen. So in some adjuvants, we use, in the old days, used to be using mercury, thimerosal, things that help stimulate immunity. And it may be that the type of adjuvant that's used in the initial inoculation for the vaccine helps stimulate longer lived immunity in some cases than others. But why some vaccines and some natural infections lead to lifelong or very many years of immunity and others have to be reboosted every 3 or 4 years? Great question, and we'd love to have the answer to that, because that's a real issue in vaccinology.
Cori, could I address a question in the
Of course.
Yeah. So one of the first questions talked about the virus mutating, and Vic addressed that as it related to vaccines. But I just wanted to talk about it a little bit as it relates to the antivirals. So, we've been trying to get a set of antivirals for HIV, for example, for years. And the challenge has been that the virus mutates so rapidly that it becomes immune to the drugs, or the drugs are no longer immune, is a poor choice of words here since we're talking about immune responses, but they no longer inhibit the activity. So what happens in those cases is that the mutations occur for enzymes that are targeted by the drugs. And the mutations make it such that the drug can't bind or doesn't inhibit when it does bind. And now that enzyme can work normally even in the presence of the drug. I would say that one of the positive things about thinking about antivirals as it relates to coronavirus is, for an RNA virus, they don't mutate quite as readily as other RNA viruses. So it may be that something like remdesivir would be more effective for a coronavirus than that kind of drug for the HIV virus. But the question asks, what changes occur? Morphology is one, so thinking about that in terms of a vaccine for like the spike protein, but it also impacts the function of these important enzymes. So the mutations can change the activity of those enzymes. So I thought that was a good question, that we can think about mutations in terms of both how it impacts vaccines and these therapeutic drugs. You're muted still.
Sorry. Another question we had is that international travel is here to stay and human pathogens will follow their hosts. How do we develop a global system to predict, detect, and prevent the pandemic spread of human pathogens?
Can I take a stab at that? I think we have to stay vigilant. I wish I could put, maybe we'll send this paper out. There's a tremendously good paper that was published in the Journal of Clinical Microbiology in 2007. It's a retrospective on all the SARS and MERS vaccines that were developed in 2004 and 2005. Most of you probably know we do not have a SARS and a MERS vaccine. We had 7 or 8 of them that went into phase 3 clinical trials, and those viruses, for whatever reason, seem to go away. We're not dealing with them anymore. They have up to 100 times more case fatality rate than this one does, but we just stopped thinking about them. That article talking about all these vaccines ends with a paragraph that says, we will see this happen again as we see the interchange of humans and bat populations and animals. They're essentially saying this is going to happen. And this was 2007. This will happen again, and we should be prepared. And we just really we have a short attention span when it comes to these things. So we need to continue to stay vigilant. The thing is that we don't know what the next virus is going to be. So ramping up a vaccine protocol against a virus that we don't know what the antigens are is a little bit of a challenge, but we have to stay focused on the fact that we know these viruses are out there. So surveillance mechanisms and collection and supporting research that will get these viruses out of the wild and understood a little bit more so that we can develop vaccines prophylactically. But it's an enormously expensive process to stockpile vaccines that we might never use.
The other thing I would add to that is the importance of basic research. A lot of research that goes on isn't immediately translated to developing a vaccine, but if we better understand the biology of a diverse group of viruses, it helps to prepare us to do the kinds of things that Vic is talking about. And a great example of that is the husband of the person who was my advisor in graduate school worked on a coronavirus called mouse hepatitis virus. And nobody really cared about mouse hepatitis virus. It wasn't causing infections in humans, but it was an excellent model to understand coronaviruses more generally. And so when the SARS pandemic, the first one occurred back in 2002, 2003, his research became very famous because it helped people be able to make predictions about this newly identified virus. And so while it may sound like it's not an important virus or that basic research doesn't, you can't immediately see the impacts on public health, it is really key to having a foundation that we can build upon as these viruses continue to emerge.
So some of it's our attention span. I mean, it's this is a big 2x4 across our foreheads, right? We're seeing what happens if we don't think and plan ahead. I mean, we've had sort of predictions in the past that something like this might happen. And it's not that anybody's not trying to be vigilant, et cetera. It's just that we have to mobilize a lot of things that we might never use in a very costly way, and it just people lose attention because we just don't think that way. So that's a real important issue.
Mm-hmm. We have about 1 or 2 minutes left. You two can see the questions. I don't know if there's one that pops out at you. If not, I can ask another one of the vaccine questions that we have listed here. It says 8 or 9 vaccines are on a fast track through clinical trials. How likely are we to see it get through phase 3? What is the likelihood that any of these viruses or any of these vaccines will be effective across variation of the virus that is likely to exist? And then another question following up with that is, should we feel comfortable with this fast speed process?
Yeah, I think we should feel comfortable. I mean, I think what's happening is rather than sequential processes to make sure we have a safe vaccine, we're spending a lot of money to do them all at once, but the safety issues are remaining at the forefront. So that's a good question, but it's not like anything, the corners are being cut. We're just doing things in a way that we would never be able to afford unless we decide to put $1 trillion into it, which is what we're doing. In terms of the numbers, it's a great question. I mean, the SARS and MERS vaccines got through phase 3. The problem was who wants to make 200 million doses of a vaccine for which there's no pathogen anymore. And so we'll probably get through phase 3 with one of these vaccines. It'll be great. And the good news is, at least from terms of public health, we're going to pay attention and we're going to keep that vaccine very likely in production because we're seeing the devastation of not having one. But if we lose our attention span again, like we did with SARS and MERS, the same issue could occur. I'm very confident we'll get through phase 3 because, I mean, the thing about vaccinology, I have a lot of friends who do it for a living. So, and if there's anybody on the call, please don't take it personally. It's not, and we've had a lot of rocket science talks. It's not rocket science. We understand the principles very readily. We know we got to get an immune response, and we're trying 160 different versions of that. One of them is going to work. Probably several of them are going to work. It's going to relate more to distribution and cost and all the other things that ultimately become problems. I think we'll get a vaccine. Unlike HIV, which has all of its own issues related to how it affects the immune system, this is a pathogen for which we should be able to develop a vaccine.
Wonderful. Cori and Vic, thank you so much. This has been a wonderful presentation. Phil, if you're still there, I don't know if you want to pop in and say the official goodbye, but thank you both for this. We really appreciate it.
Thank you, Cori. And thank you, Cori Fata-Hartley.
Yeah, thank you, everybody. This was wonderful, and thanks all for attending. We had a great talk and a great set of questions.